Go ahead.
So good morning, everyone. Thanks for being here today. It's a bit early for me, but good morning, everyone, and good night for who is like me right now. So basically, today I'm going to explain the first part of my dissertation where I try to describe the distribution of the vegetation height and density and the ground elevation in the mesotidal salt marsh using some UAV LiDAR on cloud. So first of all, I want obviously to thank my collaborators, my advisor, Dr. Canestrelli, and the other co-authors, which are Dr. Wilkinson, Dr. Itu, and Andrew Ortega. So why it's not working? Okay, got it. So basically the objective of this study, like I said before, was to convert LiDAR point cloud collected using a UAV into ground elevation and vegetation height and vegetation density. And I wanted to do it without the support of any additional data set. I want to do that because many times in the past, using LiDAR, people coupled different data set to increase the ability of this data set to determine these different features about the ground. And I wanted to skip this part using only LiDAR to detect these values. So basically the method is based on these three steps. The first thing I did was to develop an algorithm to estimate the local shape of the ground using a regression surface. And then I transformed the point cloud using the regression surface to remove the influence of the ground slope on the non-flat areas. And after I did that, I used the transform point cloud to calculate the ground elevation. And then I used a genetic algorithm and I trained it and tested using predictors coming from the LiDAR and from another data set, which is an RGB data set. And after I trained it and tested the genetic algorithm, I used the result to calculate the vegetation height and density inside my study area. This is exactly my study area, which is a highly vegetated salt marsh. You can see it here on the right. It's placed here in Georgia, close to Sapelo Highland, in a very important research reserve. And like I said before, it's pretty much vegetated with spartina alterniflora, so it's not easy to determine the ground elevation and also the vegetation properties here because, like I said before, the vegetation is pretty dense, and also because we have all these different creeks following the marsh. So to do my study, I collected different data sets. The one on the left are the one I got from a fieldwork directly on the marsh, and the one on the right are the one I got using remote sensing technologies. So basically what I did was to place some ground control points in my area. I placed exactly 27 of them. And this is an example of this ground control point. And what I did was just to take the midpoint of them using a GPS. And I used them both to georeference the RGB data and also the LiDAR data. Also, I collected some data about the vegetation here using this plot. The plot is 40 times 40 square centimeters big. And basically what I did was to take the GPS point here to get the ground elevation and also the vegetation height and density, and I did it manually. About the remote sensing data, what I did was to collect a UAV-based LiDAR point cloud using this DJI Matrix 600 UAV and with this LiDAR sensor placed here on the bottom of the UAV. And the RGB images were collected using a DJI Phantom 4 Pro drone. And these are the specifics of the flights I performed. So this is the main part of my method, basically the transformation algorithm. What I did basically was take the point cloud and to divide it in different cells. The cells have a dimension equal to the plot I showed you before, so 40 times 40 square centimeters. I call the cell PC, and E is the indexes, are the indexes of the cell, which means north and east. And basically the idea here is to calculate, to determine what is going on in the middle, in the center, in the cell in the middle, using also the lateral one. And I call these stencil, so ST and E. So what I did when after I divided the point cloud was to calculate the elevation of the lowest point in each cell, and then I calculate a regression surface using these minimum points in each in each stencil. After I did that, I calculate the distance between each point of the point cloud and the regression surface. And then to remove the effect of the slope, I just rotate each point, so the point cloud and also the regression surface with respect to the central point of the surface until the surface is horizontal. So basically what I did was to rotate the surface and then put the points at the same and maintain the distance between the point and the surface. So basically this is an example of what I did. I rotate the surface and I move the point vertically until their distance is the same we had before the rotation. And after I did that, I calculate the elevation of the minimum point, which I consider the best point described in the ground elevation. And I also calculate the relative elevation of the remaining point for consecutive analysis I did, and I will explain later. So basically why we did that? We did that just because if we think, these are two examples of what is going on in the marsh. This is basically what is going on in the middle of the marsh, in the marsh platform. You see, it is easy to say that this point, which is the lowest one we detected in this area, which can be considered as the stencil, is pretty much close to the central point and also to the elevation, as almost the same elevation of the point of the center here. So it describes very well the elevation of the central cell. But if we do the same here with the slope, we obviously have this error because the minimum point is pretty far. The elevation of the minimum point is pretty different from the elevation of the central point. So what we did basically was to rotate, to remove this effect. So to rotate this slope and to maintain this difference between the ground and the point, because we considered that if removing the slope, basically this remained the point which was detected closest to the ground. So we basically maintain the effect of the penetration of the LiDAR in the vegetation. So the first step was, like I said before, the second step, was to calculate the elevation using these points. So basically, after I collected all the points inside the area, what I did was to perform this linear regression. Obviously, the regression was done considering only the data I collected on the 77 plot I showed you before. And basically what I did was to use two different transformation regression surfaces, a planar one and a polynomial one, to see if there were some differences in the data in the results I got. And also what I did was to compare the results I got from these two methods with the results I got using the minimum elevation. So maintaining the point cloud without any transformation and using the method which is mainly used in the literature. So just selecting the minimum elevation point inside the point cloud. And these are the results. Like you can see, they are divided because I collected data on the creeks and on the marsh. And this is the interior data sets. So the interior data set, so all the data collected in the salt marsh. And you can see basically that the regression plane has the best results for the total data set and also for the marsh data set. But you see, you can see it is pretty interesting that if we use the regression polynomial curve, we have some better results here where we use on the points on the creeks. But we decided to use the regression plane in the interior area just because the marsh occupied the biggest portion of my domain. And this is basically the result from my planar regression. After that, like I said before, I used a genetic algorithm to calculate the vegetation height and density. And to train and test the algorithm, basically I used these predictors, which come from the LiDAR and from the RGB. For the RGB, I used the red, green, blue, and grayscale minimum, maximum, and mean value in each stencil. And for the LiDAR, I used the number of points, the standard deviation, fewness, kurtosis, the maximum, mean, mode, and median elevation of the points inside the stencils. And what I did after I calculate all these predictors was to standardize them. The method I use to test and train data set is this one. So basically I divide the entire data set into parts. I cross-validate the genetic algorithm using the 75% of the data set using a leave one out cross-validation method. And I use the remaining 25% to test the data set. And these are the the statistical parameters I use to basically to train and test the data set. So these are some results that I got from my formulation I got from the genetic algorithm. And I divided for the vegetation height and density, like you see. So basically, I compared the results I got using only the LiDAR predictors, the RGB predictors, sorry, this is RGB, not photogrammetry, and the LiDAR plus RGB predictors. And basically what you can see is that the error is pretty similar if we use only the LiDAR and the LiDAR plus RGB. And this is the formulation we got from the genetic algorithm. Considering these two errors, I can conclude that it's not so useful to add the RGB data. So if we add the RGB data to the LiDAR data, we don't have a very big decrease of the error in comparison with using only the LiDAR. And it's the same if we consider the vegetation density, because you see the error using the LiDAR and the LiDAR plus RGB data are exactly the same. And these errors are pretty low, considering that the maximum density we got in this area is something like 150 stands per square meter. So the error is pretty low. I didn't say before that to calculate the density, I use another variable. So basically, I pass through this sort of biomass. This is a proxy of the biomass, which was calculated by multiplying the vegetation density and the delta Z I got from the point cloud. And this is the formulation I got from the genetic algorithm. To conclude, not to conclude, but these are the results I got basically at the end of the computation for the vegetation. That's for the ground elevation, the vegetation height, and the vegetation density. And you can see here there are some zooms, which are these zooms in this slide. And basically, what I can conclude, considering these images, is that, first of all, there is a preferential sedimentation process going on here on the marsh. And you see the elevation here is pretty higher than the elevation here. And I have a theory. I just ran a numerical model yesterday, and I saw something going on related with the muscle mound population in this area. So we are going to see what is going on here using more numerical methods. About the vegetation height, we saw that the Spartina tenephora grows higher, close to the creeks and the Dublin River here at the edge, with the ground where the ground elevation is higher, and that the vegetation height progressively decreases, reaching the inner part of the marsh. Then we saw that the higher vegetation density was calculated in conjunction with the short and medium canopy height, and that the low vegetation density was calculated on the creek levees and heads in conjunction with the high vegetation. And finally, that we can determine where are these funnel shaped and low vegetated corridors used by the water to go inside and outside the marsh. Some advantages in application are related mainly on agricultural application and understanding the seasonal variation of the area in terms of the vegetation and in terms of the elevation of the area, and to verify the effect of the droughts on the vegetation. So to see if there are some patches removed by some droughts or by some hurricanes or this kind of hazards attacking basically the marsh. And then it can be used to compute some numerical observation using numerical models and basically to calibrate what is going on in the marsh. So we can use this data to see this is the vegetation we have in this area. We will use these with these parameters. To conclude, I just want to say that our approach reduces the error introduced by the non-flat ground in the computation of the vegetation characteristics and the ground elevation, capturing the large gradients in the proximity of the tidal creeks, which is a very important stuff because this is the part of the area where it is not easy to determine vegetation and also ground elevation. Also, that vegetation pattern and evolution can be analyzed using our method, obviously, and that LiDAR-derived predictors have larger predictive abilities than RGB-based predictors. and that it is better to use only the LiDAR instead of the LiDAR plus RGB dataset to avoid any additional computation effort without obtaining any increase of the goodness of the results, basically. And with this, I want to conclude. I want to thank again all my collaborators. We just published this paper in ESPL last year, so you can have more information reading it basically in ESPL. And I will be happy to take any questions at this time.